So it's time for our last speaker. It's my pleasure to invite Professor Natkunam, Professor of Hematopathology from Stanford University. And her talk is on the discovery of Epstein-Barr virus.
Thank you very much for the invitation to give this talk. The preparation of this talk was fun because I got to watch videos and read books for a change, and not scientific papers, although that is very relevant. Here are some financial disclosures. All right, when we think of EBV today, we know that it is a common herpes virus that infects most of the world. The infections are usually happens in childhood, and because the virus can establish latency, it can remain in the host asymptomatically for many years. It is also a causative agent of cancer and is classified as a biologic carcinogen. But to tell you this story, I will take you back to Africa, where a British missionary surgeon working in Kampala, Uganda, in 1957 is called after his surgical rounds to the pediatric ward by the pediatrician saying, "You're the surgeon on call and I have a case here that I want you to see." So he walks over to the pediatric ward and there sees a child with these massive jaw tumors disfiguring the young patient's face. And he had never seen anything like that. And the disfigurement as well as the discomfort in the patient was quite startling. He then, about a week later, was simply looking out of the window, which he very clearly describes, and sees a mother with a child of 2 years old, also with a similar jaw tumor. And his comment when he sees the second patient is that, one is a curiosity, but two, there had to be a cause for that. And then he starts to go to regional hospitals as well as in his clinics, where he sees patients, children especially, with tumors of the face and jaws, and he starts to record them. He goes into autopsy reports of patients, children who had died in that hospital, plus several regional hospitals, and looks at any of the descriptions that had been written down, and then also just medical records. These are highly aggressive tumors, and typically the children did not survive beyond about 3 to 4 weeks after they presented with these jaw tumors. Within a year, he had he had collected 38 cases of children with jaw tumors. He mapped the age distribution, which was presented in this first figure here, between about 2 and about 7 to 8 years of age, which is when he saw the maximum number of these cases. He also recorded the anatomic distribution, which I'll come back to. And his colleague, Professor Davis from the pathology department, also who he quotes in his first paper, he describes them as tumors that are highly malignant, with very numerous and often atypical mitoses, masses of small, dark, round cells with dark, compact, heterochromatic nuclei and scant cytoplasm, strongly resembling lymphocytes. And in some cases, the tumor resembles lymphosarcoma, which is what lymphomas used to be called in those days. So he publishes this paper in the British Journal of Surgery, which he later regrets. Of course, he's a surgeon, so he thought about sending it, but later he said that nobody read his paper and nobody knew about it. So it took a while for the idea to catch on. However, the work keeps going. Here is another description of the anatomic distribution. So even though jaw tumors were the most common to present, there were also several bilateral tumors, particularly in young adolescent children or young adolescents, where ovarian presentations were described and also skeletal descriptions. And these descriptions are quite extensive and very carefully recorded in handwritten notebooks. He then started to get referrals, and he noticed that most of the referrals were from north of Uganda and in the central parts of Africa. And his good friend, Dr. Otley in South Africa, said that they don't see this tumor in South Africa. So with that knowledge, he decided that, wow, there must be a geographic distribution for this tumor. So not very much to go on, but a good hunch. And if there is a geographic distribution, then would it be possible to identify a causative agent? Very good thinking at the time. So he applies to England for a grant, and he receives £25. And that was enough to print leaflets with some cartoons of the children he had drawn, with some photographs, micrographs, and a questionnaire, which he then mails to every hospital he can think of all across Africa and collects the data when it comes back. And the data clearly showed that the tumors were confined to the tropical regions of Africa. And there was a dependence on temperature and rainfall and elevation in the way they were distributed. So this is the original map where he is looking at this. And it's really interesting to think what careful observation coupled with, curiosity, as my, Dr. Jimenez just mentioned, could make you follow. And also that science, such good science, observational science, could be done in this manner. So then, after he gets this data, he decides that he has to visit these areas in Africa to confirm what his findings showed. So he undertakes what is now known, it is a story of legends here, as the Long Safari. So he gets two other missionary doctors. One of them is also a really good car mechanic, and this was needed on a long trip where sometimes there were no roads. And the descriptions of what they passed through and all of they visited 60 hospitals in 10 countries. It was 10,000 miles. It took them 10 weeks. And this time the research grant that he had got for this trip was a little bit more, and it was enough to buy a second-hand station wagon that they used for the trip. So his map shows where he went along tropical Africa. And at the end of it, he was able to map the disease across the malaria belt of Africa. And now why malaria belt? It's because when he came back, he showed this map to a fellow epidemiologist to try to find out. He was still after a causative agent, and he was very interested in mapping the edges of this disease to see whether, and he was thinking that it was an environmental cause at the time. And Dr. Haddocks, who was the epidemiologist that he knew locally, looked at this and said, this completely fits the malaria belt, and shows him his map. And indeed, they completely overlapped. So it was not clear, and malaria did not seem to be the causative agent of this, so it was really unclear what was causing it. Also, in the meantime, several pathologists in the area had also taken up the descriptions of the histopathologic diagnosis. And most of this work was done by Dennis Wright. This is the original pathologist whose quotes I read out that was in the original paper in the British Journal of Hematology in Surgery. And then Dennis Wright takes this up, and he uses histochemical stains and cytopathologic features to actually prove that all of these tumors in different anatomic sites, some were thought to be sarcomas, and only because different pathologists or experts were seeing them at the time. But he was able to actually, using cytology and histochemical stains, to put them together and actually prove that they were lymphomas. Then Dennis Burkitt goes back home to England for a visit, and he is invited to give a lecture at Middlesex Hospital in London. And his lecture was titled The Commonest Children Cancer in Tropical Africa and an Hitherto Unrecognized Syndrome. At the time, it was actually about the same time that Dennis Wright was working on the pathology. So the fact that it was a lymphoma wasn't entirely clear, but that was the prevalent view. Anthony Epstein was a young pathologist working on Rouse sarcoma virus-induced chicken tumors. Now, this is another interesting history. In 1911, that cancers in chicken and other animals could be caused by a virus was known. And most people thought that viral cancers, that virus-induced cancers only happened in animals. That there could actually be an oncogenic virus in humans was actually not conceived. And in Anthony Epstein's writings, he says that he was one of the few labs remaining that was still studying viruses in sarcomas and animals, and he was particularly focused on chicken sarcomas at the time. And most of the time, the field thought that we were mad because nobody believed that this was ever going to be useful for anything medical in humans. But he sat in the lecture, and about 15-20 minutes into the lecture, recognized that this unusual epidemiology and dependency on temperature, rainfall, and elevation, which Dennis Burkitt carefully describes, had to be related to a biological agent, and possibly even a virus, because he had been working on virus for a number of years already. So Epstein meets Burkitt and decides that he would come and visit Kampala to establish a collaboration to send fresh tissues. So anytime Dennis Burkitt would operate on one of these patients, he would send fresh tissue of surgical biopsies. They were overnighted to London Heathrow Airport, and whether it arrived day or night, Epstein would go pick them up and bring it back to his lab. However, for 2 years and 20 attempts later, cell cultures did not grow anything. And electron microscopy, which was the available modality to study these viruses in culture, showed no infectious agents. And this, I think, is the best part of the story. In late December 1963, due to very foggy conditions and bad weather in London, the flight carrying a biologic sample could not land, and it was diverted all over the place, and eventually, very late, arrived at the Heathrow Airport, many hours, in fact, almost a day late. And so Epstein, of course, religiously goes to pick it up. And he sees that there is fluid around the sample, which is cloudy, and which is suggestive, of course, of bacterial contamination. And it was going to be discarded, but he thought, okay, let me take it to the lab anyway, met with his graduate student, Epstein-Barr, and they looked at the sample under the microscope. And lo and behold, they found free-floating tumor cells. Now, that observation was fantastic because he also realized he had been trying to culture these viruses on plates and other types of media. Instead, he realized that he should culture this sample in suspension, which he did. And the cells proliferated and became a cell line, the first EBV cell line, which they called EB1. And within weeks, they had enough cells to do electron microscopy. Now, another pathologist working with Epstein, Bert Ashong, who is a Trinidadian pathologist who was working with him, he was an expert electron microscopist, was able to find the virus particles. So this is from their paper, which was published in Lancet in 1964, where viral particles, which are shown here in the white arrows, and you can clearly see them in the first electron micrograph that they produced. Now, for his contributions to the field of human viral oncogenesis, Anthony Epstein was later knighted. Bert and he worked together for many long years, and Epstein-Barr, who characterized many of the characteristics of the virus, also received her PhD. Okay, so then once you discover a virus, or they didn't know, whether it was, but the viral particles were the proof, they sent it off to a virology lab that was run by a husband and wife team, Werner and Gertrude Henley, who were working at the Children's Hospital of Philadelphia in Pennsylvania. And there, they were able to use known serologic agents from other sera, from other human infections, to pinpoint that this was indeed a novel virus, and it didn't have the reactivity pattern of any of the known viruses. And they named the virus Epstein-Barr. with the eponymic name that it is known for, or it is also called human herpes virus 4. And then, again, with incredible insight, Epstein decided that if this is a virus and it can cause cancer, it's going to need a vaccine, and it's going to need an animal model to study this further. So as early as 1970, he actually created one of the first animal models in New World monkeys. Much had to happen before, and here is a timeline of other milestones of EBV. So not only was it the discovery in Burkitt lymphoma cell lines, and then also showing that it was present in tumors directly, but in nasopharyngeal carcinomas, gastric carcinomas, the genome of the EBV was detected and sequenced, et cetera, et cetera. I won't go into all of these steps. And in 1997, the International Agency of Research on Cancer then classified this as a group 1 carcinogen, so which means that it's directly causally related to oncogenesis. And these are some of the first papers. You can see the Burkitt lymphoma nasopharyngeal carcinoma paper as early as 1970, where they show it actually in the tissue. T-cell lymphomas containing EBV DNA, the DNA sequence of the first isolate that was actually identified the EBV genome, and then DNA tissue in Hodgkin cells and in gastric carcinoma. So I just wanted to highlight the paper. So this happened over the next 30 years. Today, we know that the EBV viral genome is actually completely sequenced, and how many reading frames it has. It's a double-stranded DNA virus that is transmitted through saliva, and that is why it is easy for anyone to get infected. What is unusual about EBV and why it is persistent is, in fact, because it has this dual phase life cycle. So it can establish a lytic infection, but more commonly, it establishes a latency program, which has many steps, and the depth of the latency can actually evade the human immune system extremely well. And because of this, there has been a lot of study going into this. But because of this, even though it causes only a self-limiting disease, most of the time, it can actually persist in the host in a lifelong once it infects the host. Acute infections in teenage and young adults also happens, and this is now known as infectious mononucleosis. Originally, it was described by the couple that I mentioned previously, the Henleys, and they called it glandular fever. This is also mostly self-limiting. It can cause some acute complications, as you know, such as splenic rupture and so on. And there are also long-term complications, which I will talk about. There are now also, over the years, models of how EBV infections happen in B cells by the directly virion infection, as well as in epithelia, where it can be passed from cell to cell. And in addition to lymphomas, many of the lymphomas that are listed here, there are also smooth muscle tumors, gastric and nasopharyngeal carcinomas, but other immunopathologic disorders, including the latest one that was proven as multiple sclerosis. Okay, so biological carcinogens, I'm not going to go into this, I will spare you all this, but simply to say that the pathways that activate after EBV, so its genome is known, LMP1, latent membrane protein 1, is also known as the major oncogene of this virus, and the signaling pathways and how it transforms cells has also been worked out. And although this is the first human oncogenic virus recognized, there are many others. Today, it is estimated that over 200,000 new cases, so this is incidence, not prevalence, 200,000 new cases of cancer are caused annually by this virus, and I have listed some of them here. Okay, and then the patients who are most at risk are those who are immunosuppressed. So I told you before that EBV persists in the memory B cell pools of infected hosts, and this is lifelong. EBV encoded latent genes can then induce transformation. And in most immunocompetent people, this doesn't matter, and the host and the virus can live happily for a long time. However, once a patient is immunosuppressed, and there are many causes of immunosuppression, which can be innate, like in congenital, innate immunity and genetic disorders that cause immunosuppression, drugs or transplantation in the iatrogenic categories, as well as HIV. And in the other end of life, you also have immune senescence, and there are many of these situations that may not be apparent because transplant and maybe even HIV, if it's detected, is apparent, but there are many other causes of immune suppression that may not be. And it is those patients that typically develop EBV-related lymphoproliferative disorders. Now, the discovery of EBV also led to lots of other work, and now globally up to 20% of cancers are attributed to viruses, and this is only a partial list. And then the role of viruses, it also started inquiry into, is it really causative or is it a cofactor? Are there other epidemiologic factors that influence how the cancer is created? So what is the oncogenic pathways? And then are there altered genetics and host immunity that also take place? And that brings me to why the original map, Burkitt, completely overlapped with the malaria belt. This is because, and this was described only about 10 years ago, so it remained a myth for a very long time, but this elegant paper from the Nuschenschweiss group actually showed how chronic Plasmodium-infected germinal center B cells show increased AIDE activity, and AIDE is the enzyme in the germinal centers that is responsible for the transcriptional machinery that causes somatic hypermutation, et cetera, in the immune response in the germinal centers. And this permissive environment that is created by Plasmodium infection, and particularly because in the laboratory they could show that if they increase the parasitemia of infection of the germinal center B cells, you could actually increase AIDE activity. And because AIDE increases DNA damage and translocations, including MYC, immunoglobulin translocations, which is really the oncogenic part of the cancer, this actually provides a perfect storm along with EBV, which has the transforming genes, and MYC, which is the master oncogene, to create a situation that you have more risk of developing Burkitt. So the primary EBV infection also occurs in very young ages in sub-Saharan Africa. So there are many reasons why this early childhood patients in their early childhood develop these, because this permissive environment gives a much more enriched environment for oncogenesis. Now, EBV-induced tumors happen worldwide. They are not particularly, even Burkitt lymphoma is not particularly restricted to young children. As we know, it is sporadic and occurs worldwide, but this is now called endemic Burkitt because of its association with malaria. There are also now there are two main strains of EBV that is known, and I just want to show you a phylogenetic classification that it is geographically dependent. It is good to know that this exists only because there are attempts to still make vaccines, and the geographic distribution of the differences in the phylogenetic tree may make a difference in how and where vaccines are rolled out. In fact, a couple of EBV vaccines are in clinical trials, and this is something that might become very relevant for future vaccine distribution in the world. So I hope I have given you a snapshot of a serendipitous discovery that actually led to the identification of a human oncogenic virus. It set the stage for studying human viral oncogenesis in general in lymphomas as well as in other cancers. The first chemotherapy for lymphoma was also pioneered here. This is a story I didn't touch on because this is part of what Dennis Burkitt managed to do. Sloan Kettering Cancer Institute had an investigator who was at that time, had pioneered several drugs. And he actually offered methotrexate for highly proliferative tumors. And the first African child was actually treated in the kitchen. And Dennis Burkitt did not know how to give chemotherapy, but he learned it from his collaborator, and he gave it. Now, there are lots of things you could say about how that would just be an absolute no-no in today's world. But within the first 10 years of the discovery from identifying jaw tumors in a young child, the next 10 years, what he was able to achieve along with some very insightful colleagues has been tremendous and just opened up the field for very many investigations. Now, we know a lot about EBV, but there are ongoing challenges, including developing an effective vaccine. It has not still happened. I say effective. There are some vaccines that are in trial. Understanding the host immune mechanisms, biomarkers for early detection of cancer so that preventive strategies can be meted out to vulnerable patients. So this actually may become curable, and patients, oncogenesis in the background of viruses, if viruses or biomarker could be detected early, could be a way that this can be mitigated. Thank you.
Thank you. Are there any questions from the audience, please?
So is there any thought on why the endemic cases of Burkitt lymphoma are in the jaw primarily, whereas sporadic cases tend to be in the abdomen and other locations?
Yeah, great question. Actually, I think it's in his writings in the early, which was the most dramatic presentations, but the localization to jaw tumors is not very clear, but it does have an unusual distribution to other sites like, puberty, children in puberty getting bilateral ovarian cancers that are Burkitt and the like. So it does have an unusual distribution, which is also something that made it stand out when people like Anthony Epstein heard the presentation. Exactly why it homes to jaws is not really clear, but it does affect long bones, which is the other place that young children get these tumors.
Very nice talk, thank you. When the infected cells were cultured in suspension, they were sent to electron microscopy. You showed in the slide there are 3 individuals, Epstein, Barr, and Parker.
Chong, yeah.
What happened to him?
He worked with Epstein for a long time. Unfortunately, Burkitt did not want this particularly named after him. And he would have also he has actually mentioned that it should have been Burkitt Epstein-Barr virus, but he didn't name it. Other people named it for him and his graduate student at the time. That was a misnomer, and he acknowledges it because the electron microscopist who worked with him for many years to come, they worked together and published many papers together, didn't get the eponymic thing was applied, not by them.
Just as a comment, Burkitt's lymphoma is very common in greyhound dogs as well too, and that's one of the few times that I've seen it in a veterinary specimen.
Interesting. Yeah, that's a really nice comment. Thank you. Dogs can be a very good model for lymphoma. I have seen diffuse large B-cell lymphomas in dogs. I have not necessarily looked for EBV in them, but they are a good, it's probably the best animal models for lymphoma, and I've heard that from investigators working in the field, but that's very interesting.
2 more comments. I think Burkitt also, on his travels, came to a village where malaria had been eradicated, but there had previously been a French missionary surgeon there who was a very skilled sculptor. And in the closet where the French missionary had lived, they found statues of Burkitt lymphoma children, models of them. But
right.
malaria was gone, so, and there was no more Burkitt in the village. These were from years ago. And the second comment is, in later life, also part of his Africa experience, when he came to villages, he also noticed that there was virtually no colon cancer and no polyps. And so he became a big proponent. The last part of his life was going around the world proposing high fiber diets.
Yes, that's exactly right. Thank you for your comment. And his next major contribution was about the epidemiology of colon cancer. He went back to, Uganda got its independence, and the medical service contracts for the British, and Irish servicemen there, including the medics, was ending. The long safari actually was undertaken in a rush in a way, because he was worried that he would not be able to complete his work before he left. But when he was back I think he worked in Gloucester after that, in England, he did, again, careful annotations of and record keeping of patients' dietary fiber and colon cancer. And he spent the rest of his life actually writing and researching that. That's absolutely right.
So thank you, Professor Natkunam. And just a few words to conclude the session. I wish to thank the speakers for their inspiring, brilliant talks, all the participants of our companion meeting, and personally Dr. Mary Christine Aubrey, for not only for being moderator, but for her support to the History of Pathology Society activities. And I invite Dr. Barrios to join us on the stage to receive the gavel, which marks officially the transition of the presidency. And
Good job. Thank you very much. Thank you.
So that is all. Thank you very much for attending this session. Please, if you're interested in learning more about our society, we do have a webpage. It's just called the History of Pathology Society.